Hello, everyone, and welcome back to chapter 14, part 2. So, in the last part, we spoke a lot about DNA. So, what is this, genetic material that's being passed on from one organism to the other that, we inherit, the traits and everything. So, we were really discussing a lot of history in what, made us believe that it was actually DNA and not protein that was actually being passed on. And then after that, we spoke a lot about the structure of DNA, that it's actually two strands, anti-parallel, meaning one's going from 5 prime to 3 prime, and the other one's going from 3 prime to 5 prime. And they're basically, having this really nice, they're binding together in this really nice alpha helical kind of complex. And this is really basically really cool. And we understood that this is a very nice structure, but we didn't know during those times how did this big and nice structure like the alpha helix was able to replicate. So was it actually being done in a sense that, a new whole DNA strand is actually formed completely new and the old strand stays as the same? Or is that, when this old strand, what happens in this con is that the duplication actually gives you two DNA, one strand that's old and one strand that's new, or the dispersive model in the sense that no, basically when you duplicate, both strands are made of both mixed, like a half a mix of old and new. So you can see that this strand over here is made of old and new, and this one as well is old and new. And then basically, Massillon and Stahl were the ones who actually showed that no, actually, it's the semi-conservative model, and where when we duplicate DNA, we actually have a new DNA strand formed that is half old and half new. So now let's actually talk about DNA replication. So with DNA replication, there's three things that you require. You require something to copy. So the parental DNA. So in this case, you need this. You need basically this to copy because you need to basically separate these and then you basically copy from that. Then you also need something to do the copying. So a protein that's doing the work, hence an enzyme, which is the DNA polymerase, which we will talk more in details about it. And of course, you need the building blocks. So these are the monomers. You need nucleotides to basically build you the new strand completely. So that's what you need. Three things: the parental DNA molecule to copy, the enzymes to do the work, and the building blocks, the nucleotides that are building the new polymer of DNA strand. So the stages of DNA can be actually, put in three stages: the initiation, elongation, and termination. Initiation is basically when replication happens. This is the start. So in this case, actually, what happens, the DNA is kind of open up, or is kind of broken and opened up. So it's, the DNA is double helix, and then it's gonna basically be, broken into each one helical strand, so one strand and the other. And then you have the elongation. This is where now the new DNA is being synthesized. So the DNA polymerase, so this enzyme is adding new blocks to create this new strand. And the final part is termination, is basically when replication is terminated, which will go into every single one of these stages more into details. Okay? So let's talk about more the action of DNA polymerase. DNA polymerase is actually the enzyme that's doing all of the synthesis of this new actually DNA strand. So the good thing about this thing, well, the good thing, let's say one of the negatives of DNA polymerase is that it's only able to create nucleotides to the 3' end of the growing chain, meaning that it actually puts DNA from the 5' to 3' of a new strand. So here you have basically a strand that's 3' and 5'. So it's able to actually put it. But if this was 5' and 3', it would be a bit different. So again, DNA polymerase can only add nucleotides to the 3' and end of a growing chain. So it only adds DNA from the basically 5' to 3'. So it only adds it to the 3' basically growing chain over here. So this is the growing chain, this is the 3', and it will be able to give it because basically when it actually creates the new um, strand, it creates it from 5 to 3'. And then, of course, it uses nucleotides as your polymers. However, it uses these kind of adenosine triphosphate. So sorry, these basically nucleotide triphosphates. So these nucleotide triphosphates. So we know. Let's go back actually to the here, maybe a bit more here. So this is basically your nucleotide. It has only one phosphate group, right, in the 5'. Well, the building blocks actually have three. So they're actually nucleotides triphosphates. And what happens here is that this bond over here is broken, and this breaking of the bond, as we know, the phosphate bonds, like an ATP, remember, have a lot of energy. This is what the energy is getting it to, so, to do this, actually, this whole um, building of a new DNA strand. So when you basically break these bonds, because we don't want, when you put this, you see you only have one phosphate. So you break these two phosphates away, you break this gives you energy to build this. Because building this is actually an endergonic reaction, a reaction that needs energy, right? So what we do is we break these double phosphate bonds in order for this guy to basically now bind to the OH group, the hydroxyl group, and continue with the strand. So that's how DNA polymerase works. So again, let's go back into this. DNA bases will only add complementary nucleotides. So for example, if this is A, over here, it will only build you a T. If this is G, it will only give you a C. So this is C, this is a G. This is a C, it's going to be basically a G. So it only basically add nucleotides that are complementary to each other to make sure Chargaff's rule is fulfilled, meaning that the number of A is equal to the number of T, the number of G is always equal to the number of C. So DNA basically polymerase will add DNA bases with complementary nucleotides. And as we know, we spoke a lot about these common features. It only adds bases to 3' ends of existing strands. So it will, from the already existing strand, it's only gonna put it in the 3' end only, and it synthesizes from the 5' to 3' direction. That's it. It cannot do the opposite. And this will become a problem because remember, guys, DNA. So if this strand, so if we remember the parental strand, this if this is 3' to 5', the other ones could be 5' to 3'. So DNA polymerase cannot do the same thing. So we'll talk about this a bit later. Okay. So again, it synthesizes in the 5' to 3' direction. So you have this new, basically the old strand over here. That's 3' to 5'. It adds to the 3' end, and it creates a new, basically, strand from the 5' to 3'. However, this DNA polymerase cannot add nucleotides to a completely free strand. This free strand has to have a primer that's made of RNA. So this yellow actually arrow signifies a RNA primer that another enzyme does, which we'll talk about. What if this enzyme does? It just creates this small little primer, which is basically around 10 to 20 nucleotides long. So it's going to create it. And then DNA polymerase is able to recognize this, and they're like, okay, now I can build on top of it. Because if this doesn't exist, the DNA polymerase cannot do its job. So it requires a primer of RNA, because this thing will tell it, it's like, okay, now you can actually build upon it. So as you can see, in this case here, this is the RNA primer that's binding here. This, the polymerase shown here in red comes in, and now it can actually synthesize the new one, which is shown here in blue. So this is the new strand that the DNA polymerase does. So this basically is RNA, and this is DNA. So this one actually, we need to remove it. And we'll talk about this a bit later. But I'm just telling you more the characteristics of DNA polymerase. So now we're going to actually here just talk about the prokaryotic replication. So prokaryotic replication is just replication done in unicellular organisms. And we're going to talk a lot about E. coli. His E. coli is a beautiful model. It has a singular circular DNA. So its DNA actually looks like this. It's one big massive circle that's closing on itself. And it actually, the replication of DNA in E. coli is super fast. We're talking, it actually can go 1,000 nucleotides per second. It's crazy how fast it can go, actually. So imagine, and you need it because imagine the E. coli in the perfect conditions can replicate every 20 minutes. So you need actually to DNA for it to replicate really quickly and accommodate for this. And the nice thing of these actually DNA, this E. coli DNA in prokaryotes, they have something that's called an origin of replication, shown here basically in blue. And they also have something called the region of termination, which is shown here, basically this region of termination here in red. So the origin, this origin of replication, the name is very self-explanatory. This is where actually DNA replication occurs. And this origin of replication is heavy in A and T. So adenine and thymine actually nucleotides. It's a very heavy region with a lot of A and T. And then you might start thinking, why is it heavy in A and T? Why is that the case? Well, it's quite easy actually. Because if you remember, A and T have double hydrogen bonds. GC have triple. So if it's a big portion of A and T, it actually is easier to open compared to if it's a region to GC. Because imagine if it has a big GC content, it actually become a problem because it's super hard to actually open it. But because it's A and T, it's much easier because only it only has two hydrogen bonds. So what happens here is the origin of termination is basically where your DNA polymerase and many other enzymes that we're going to talk are literally going to come in. And this is called the replosome. This is literally where all your actually DNA synthesase. So these are all of these kind of enzymes that are helping in DNA replication come, and they start to actually go and replicate in both directions. So this direction over here and this direction over here. So this one is going to actually take the bottom strand, and this one's going to take the actually upper strand, and they're going to keep on going on and on and on until you reach the termination point. So the termination point, as the name says, is basically the point where DNA replication ends. And that basically helps then here the enzymes are recognize this and they cut it into so you get two copies. So this is basically how the prokaryotic replication occurs in a very easy manner. Okay. So one thing that I also want to say is that a DNA, a piece of DNA with an origin is called a replicon. So that's what it is. So in this case here we have one replicon because it's one DNA with one origin. Here we have two replicons. It is one DNA with one actually origin site. So imagine if we had one piece of DNA and it doesn't have a replicon. Sorry, it doesn't have a origin of replication. Then it's not a replicon. It's just a piece of DNA. Replicon is literally DNA with an origin of replication. That's what it means. Keep that in mind. So in the case of a circular chromosome, like this one over here, it actually has one replicon because it's one piece of DNA with one actually origin. If this, for example, had another, let's say, origin, you would actually have two replicons because it's another piece of DNA with an origin. So now let's talk about E. coli and their polymerases. So first, scientists thought that E. coli has just one DNA polymerase, which they called it DNA polymerase one. That's what they thought. But no, actually, it has three DNA polymerases. So it has DNA polymerase one, two, and three, which you can also call them in short pol one for DNA polymerase one, pol two, and pol three. So DNA polymerase three is the main replication enzyme. And DNA polymerase 1 actually also is required for replication, but it actually more is helping on the lagging strands to remove primers and replace them with DNA. And we'll talk a lot about this a lot. The DNA polymerase 2 is not that actually required that much in DNA replication. It has a big function when it comes to DNA repair processes. So where you really need to repair certain DNAs because they got a mutation or something is wrong. And we'll talk about this a bit later. However, all of these guys, so DNA polymerase 1, 2, and 3, have this 3 to 5 prime exonuclease activity. So what does this mean? Is that these enzymes are able to cut, they're able to actually eat up nucleotides from a 3 prime to 5 prime actually direction. So let me show you here. For example, a exonuclease, it's basically able to cut and misplace the bases. So exonuclease means that it can only, so go back here, sorry, cut DNA in its ends. So hence the name exo. And it cannot cut it here. So for example, the DNA can come here and actually cut it again from 3 prime to 5 prime. So it can only cut this one here. It does not cut this because this is 5 prime, 3 prime. No, it needs to cut it from 3 prime to 5 prime end. So it's able just to cut these area. That's what's able to do. So that's exonucleases. DNA polymerase 1, it's a bit more special than the rest because it has an extra activity that's able to also bind to the exonuclease activity. It has this 5 prime to 3 prime exonuclease, meaning that it's able to also cut basically this part over here. So this DNA is able to cut it from 5 prime to 3 prime. So it's able to do so. So that's only DNA polymerase 1. And this is very important because it's able to remove RNA primers. This a guy here, the 3.5 prime, it's able to do proofreading because DNA replication happens in this direction from 3 prime to 5 prime. So if the polymerase did something wrong, it's able to go back up and basically bite it, like, and remove a bit. This guy, no. This guy, for example, like 5 prime to 3 prime, DNA replication doesn't occur from that. And this is more for removing RNA primers that we spoke about. We'll talk about this more. So now let's start actually with initiations. The initiation first starts off with having DNA unwind, because DNA is actually again made of two strands that are both, doing this nice alpha helical structure, right? So we have this enzyme called helicase. So helicase is an enzyme that's able to unwind DNA. So as you can see, this is DNA over here that's having this really nice kind of alpha helical structure. So this helicase, as you can see here, is opening it up. So you see now it's this opening this strand over here. So it's completely opening it. And then of course this uses ATP. To open up, you need ATP to break these hydrogen bonds over here. So of course, when you open these up, the issue with these strands that they're very hydrophobic, the nucleotides inside are very hydrophobic. So they will try as much as they can to repel back inside. So you don't want that. So what you get is the single strand binding proteins, so SSB, which are literally coating these strands. So these basically now open strands, they're literally, binding on it. And what they do is they prevent the two strands to come together. That's what they do, these single strand binding proteins. So they keep them apart. So I'll repeat this. Helicase, what it does, it actually opens these double strands, completely opens it. And basically, of course, now these, I see this one here is open and this one is open. To keep them open and not these to go back, because these are very hydrophobic and they would love to get together, you have these single strand binding proteins that are literally binding over here and here and preventing to bind again. So one thing that also happens, because remember, this is basically all wound up together, right? And what happens is that when you open this, you will create some basically this topiometry stress, topoisometric stress. So it's literally the stress that's causing these coils. Think about this as if you have literally a rubber band that's literally twisted on each other. So you have two bands are twisted on each other, and if you open it, once you open, the other parts are gonna even twist even more. That's called in DNA supercoiling. So when you see here now, this is opening. This DNA here is getting coiled a lot because you're putting a lot of pressure, and this is called topoisomerase pressure, basically pressure, and you're really creating these really super big coils. So you don't want this. So what you're going to use is topoisomerases. These are enzymes that are preventing this supercoiling. And in DNA replication, it's called DNA gyrase. So you can see here now, for example, here you don't have any DNA gyrase. You're getting these really big supercoiling happening because of the helicase that's unwinding. So you're creating this topoisomeristic pressure. When you put the DNA gyrase, it's preventing now this supercoiling. So DNA gyrase is a topoisomerase. So topoisomerase is just a type, and one of them is DNA gyrase that's preventing basically these supercoils. So that's the initiation. Now we've opened this up, we've prevented these strands basically from binding to each other, and we prevented actually now these supercoils. So now we can actually, move on. So now replication is semi-discontinuous. So this is what it means. DNA polymerase can synthesize only in one direction. Remember, we spoke about this. If you go back here, it only can go from the 5 prime to 3 prime end. So it can only put DNA nucleotides in the 3 prime strand. This is a bit of an issue, don't you think? Don't you remember that DNA is two anti-parallel strands? So one of them is correct, that is 3 prime to 5 prime, but the other one is 5 prime to 3 prime. So it cannot actually add a complementary strand that is 3 to 5 prime because it doesn't do that. So what happens here is you have a leading strand that is synthesized continuously, and you have a lagging strand that is discontinuous. You have multiple priming events, and we'll talk about them here. So this is your piece of DNA, okay? So when you open this, you're going to have basically one strand that's 3 prime to 5 prime. And then you have this one that is basically, 5 prime to 3 prime. This one is perfect because now basically what we have to do is just put an RNA primer over here. So we put the RNA primer, and now basically DNA polymerase 3 can come in and do the job. DNA polymerase 3 is able to actually continue here in the 5 prime to 3 prime direction. So it's able to add nucleotides in the 3 prime and 5 prime strand. So remember, it's putting nucleotides complementary to 3 prime and 5 prime, but it's nucleating, it's creating it and basically from 5 prime to 3 prime. Do not confuse this, please. However, here, this is a bit of an issue because now you have it's not able to do so because this is basically, um, the 5 prime to 3 prime. It cannot now put nucleotides in the 3 prime to 5 prime end. It can only put it in the 5 prime to 3 prime end. So the strand that is 5 prime to 3 prime, it's creating basically an issue. So this is your actually lagging strand, and this is your leading strand because basically this will continue on in one go, so continuous with no problems. This whole enzyme, the DNA polymerase 3, is going to go with not a problem. In the lagging strand, you need multiple RNA primers. So you need one here, and now when you put one here, you can actually go from here to here because now this is 3 prime to 5 prime. Sorry, so this is 3 prime here and 5 prime, so it can go this way. But it cannot go this way anymore. It cannot go back. So you need to now go again, put another primer here, and go again, and multiple. So you create multiple DNA fragments. So as you can see, you have one DNA fragment here and one DNA fragment here. Because the first one, it's not okay. You can basically add it here, then you can just add from here to here because that's the only way DNA polymers can go. But this now DNA that's open up, you need to put a second primer, and then it goes again. So you create these DNA fragments. And these DNA fragments are called Okazaki fragments. And their name comes from the scientist that was able to discover them. He discovered these multiple DNA fragments that are coming actually up in the lagging strand. So these are called Okazaki fragments. So as we said, we have a leading strand that's one go and a lagging strand that is discontinuous. So you have multiple RNA primers and multiple, DNA, basically, parts of DNA being synthesized. So one here and another one here, and if this continues, you'll have more and more and more RNA primers. So now let's talk about actually this RNA primer. This RNA primer is actually given by a enzyme called DNA primase. So DNA primase is an RNA polymerase actually, because it literally its name makes RNA. So it makes the RNA primer. So first thing that has to come is that this DNA primase has to first come here and create this RNA primer. And same thing here. For this one, for the leading strand, you just need one, and this continues on and on and on. For the lagging strand, you need DNA primers to give you multiple RNA primers until the whole thing is done, and you create these Okazaki fragments multiple times. Later, DNA polymerase 1 is able to remove these RNA primers. Then you start thinking, but why doesn't DNA polymerase 3 do that? Why? Because as we said, DNA polymerase 1 has this 5 to 3 prime exonuclease activity that DNA polymerase 3 doesn't have. So DNA polymerase can cut from 5 prime to 3 prime ends. So it basically comes here. So this guy over here is basically 5 prime to 3 prime. So this is 5 prime to 3 prime. This is 5 prime to 3 prime. DNA polymerase 2 cannot cut it. So you need DNA polymerase 1 to come and cut this. So this nucleation shown here basically in blue is done by DNA polymerase 3. These RNA primers are cut by DNA polymerase 1, and what happens, it basically then connects these two together because it can also create nucleotides. So it connects these two Okazaki fragments together, and this connects this together. So that's what DNA polymerase 1 does. It removes these RNA primers and also joins the Okazaki fragments. And these basically now, when you remove the RNA primer, you have these extra nucleotides that you need to create. So in the leading strand, it's very beautiful, actually, this how this happens because literally this big DNA polymerase 3 literally binds here and it goes continuously. It just keeps going in one go. And if you look actually at DNA polymerase 3, it's a very big multi-protein complex. So it's more made up of many different types of subunits. And it actually has a subunit called the beta subunit that literally looks like this. So it's literally two beta subunits, as you can see here, that form basically this ring. And what it does, it literally clamps. So it literally closes up like a clamp on this DNA and this DNA molecule, and it slides. It will just purely slide like that in a way that it will keep going to the strand in a continuous fashion. And that we also call processivity. Processivity. Processivity means that this enzyme is able to go in one go and basically do this nucleation in one go. That's the beautiful thing of it. Because it literally has this beautiful structure, this beta subunits that act like a ring that clamp onto DNA and they slide through the leading strand. That is super beautiful. Actually, I've seen these pictures, actually some videos about it, and it's that enzyme goes like freaking crazy. It's super fast. So now let's talk a bit more about the lagging strand synthesis. So the lagging strand, as we said, you have basically this discontinuous synthesis. So you have basically DNA polymerase three. That's just like the leading strand. The DNA polymerase three is creating these kind of blue stuff over here. You need primase because you need basically these RNA primers. So you need multiple of them, as we see here. So we need multiple RNA primers, so you multiple of them. And then basically what's going to happen is DNA polymerase three is going to recognize this and build over here. And then it's going to stop. Then it's going to come back here. It's like, oh, another one. And it's going to build here and it's going to stop because now there's this guy over here. So then it stops. So you're creating multiple Okazaki fragments. DNA actually polymerase then removes all the primers and replaces the DNA. So DNA polymerase 1, it basically comes in here and removes this thing over here, and then it basically builds the nucleotides and removes this and builds the nucleotides. The final enzyme you need for this discontinuous synthesis is DNA ligase. So the name is quite self-explanatory. It ligates. So now basically DNA polymerase removes this and creates this. It needs to connect this now to this strand because this strand over here and this strand are not connected, and DNA ligase creates this nice now actually um, ligation. So it connects these strands together. So it connects the Okazaki fragments together. So this is the lagging strand here, basically. So as you can see, this is the lagging, this is the RNA primer. You see here now this DNA polymerase over one. It's removing basically these now, what do you call it? This RNA primer, it's removing it. And you have this DNA ligase. So as you can see, this one here, this is not connected to this. So now this DNA ligase is connecting it now over here. So that's what the DNA ligase is doing. So you can see the DNA ligase is not, this is an Okazaki fragment, and this is another Okazaki fragment, and it's connecting them together. This DNA polymerase here, as you can see, is removing basically these RNA primers and adding nucleotides. So when it's going to add nucleotides, it's going to convert this yellow to blue, and then this blue is going to be connected to this blue by DNA ligase. And you can see here, primase is what it's doing is literally actually just adding more primases. As more you open this, you need to add more RNA primers, and that's your primers that's doing it. And this blue is being done by DNA polymerase 3. So now comes the ending, which is basically the termination. This occurs at a specific site that we spoke about in actually E. coli. So you have basically this termination down here shown in red, and this kind of tells basically all your nucleotides like, okay, stop and don't do anything, and it stops. And usually, of course, makes sense. It's located roughly the opposite of the origin of replication. So if you go back here, this is the origin is literally roughly on the opposite side. So the termination is literally opposite of each other. So the last stage of replication produces two daughter molecules that are intertwined like two rings in a chain. So you really get two rings in a chain, of course, because if you see here, if you go down here, they're gonna comply with each other, and you're gonna have two rings connected to each other. So you need something that's gonna cut this, and who's gonna do this is DNA gyrase. So basically, these intertwined molecules are unlinked by the same enzyme that relieves the torsional strain in the replication fork. So DNA gyrase, it's gonna come and basically unhook these two rings that are connected to each other. It's gonna unhook them and give you two nice strands, and that's termination. So this is basically how you actually have DNA replication in prokaryotes. Next, we're going to talk about eukaryotic replication in our next video.